Sente? Mi sente?
Stefano Colmi. Iniziamo? Siamo agli ultimi 10 minuti accademici. Io sono molto Good morning, Frédéric. Piacere you presence in Fano. This is the second lesson of the fifth day of teaching week from University of Bourgogne. And the lesson of biomineralization for that origin, evolution, the genetic process for your expertise in these topics. Iniziamo la lezione, è registrata, recorded. This lesson is recorded because the student and possibility of fill the question on the report that you prefer. Grazie, Frédéric. Please.
Thank you, Mauro. Before I start, I would like to thank Mauro and Stefano for giving me this opportunity to give this talk. So, first of all, can you hear me very clearly? Is it okay?
Yes.
So, because I like to move a little bit, you know, when I present. So, but I will try to shout when necessary. So today we are going to speak about biomineralization. So I suppose that most of you know a little bit what it is, but I will try to convince you that this is a very important field in science. So I will start. I will have like 4 or 5, depending on how long it will take. But I have like 5 different presentations, one after each other. The first one is an introduction. And shall I start? Okay, so here. But just before I start, I just present or introduce myself very briefly. I'm coming from earth science field, but already during my second year of master, I worked with bone collagen. So it was not completely earth science, but something going to biology. Then I did a PhD thesis at the Université, Paris 11, in paleontology, and I worked a lot with biominerals, so calcium carbonate biominerals. Then after I did my military service, at that time it was still existing in France and probably in Italy too. So it was something that had to be done. And after that, I made a first postdoc, then a second postdoc. So I emigrated to the Netherlands, to at Vlakkeland, the flat country, where I worked at the University of Leiden for more than 6.5 years. Then I worked in a private company for 2 years, located in Holland too, close to Utrecht. Then after that, I was recruited by CNRS as a researcher. I got my habilitation in 2009, and then I became a research director at CNRS a few years after. So, just to present very briefly what we do in Dijon, we are really a transversal research unit. There are some people, some researchers working with ecological problems, like relationship between parasites and their host, okay? But also some people working with climate. And in between, you have this Samba team. And in the Samba team, there are 2 permanent people working on biomineralization. So that's my colleague, Irina Bundeleva, and I. And my colleague worked mostly on bacterial biomineralization, bacterial calcification, while I mostly work on metazoan calcification. And since 2004, we have 8 PhD theses in biomineralization, and the last one is Camille Lutetoti, who will normally receive a PhD in about 1 month. So now let's start with the real deal. When you speak about biomineralization, there are different meanings. First, biomineralization are mineralized structures which are produced by living systems, okay? But when you speak about biomineralization, you also speak about a process. So biomineralization is a process by which living systems make minerals. And finally, biomineralization is also a discipline, a scientific discipline. So now in these few coming minutes, I'm going to focus on the discipline aspects of biomineralization. Biomineralization as a discipline is really at the crossing between many different topics or many different fields, scientific fields. I write here on this slide all the fields which are, let's say, on the upper part, all the fields which are really fundamental, like biochemistry, molecular biology, physiology, cell biology, crystallography, mineralogy, archaeology, paleontology, evolution, sedimentology, geochemistry, and in general sense, more generally, environmental sciences. But there are also some fields which are much more applied. It's aquaculture, pearl culture, zootechnics, biomaterials field, health, bone and dental surgery. So biomineralization is really at the crossing between all these fields. And it's interesting to do a little bit of history of this discipline, because it started actually quite early. First remark is that already for a very long time, human beings used biominerals for all kinds of purposes. And for example, show the example of the oldest ornaments which were found in the cave in Bismoun in Morocco. And in this cave, the archaeologists found these little shells which were pierced and which were used as ornaments. And that's the very first use of biominerals, biomineralization as ornament. So that's 142,000 years ago. Closer to us, we have the very well-known example of the shells which were sewed on clothes, which were also used as ornaments. And these are the Grimaldi children, 11,000 years from now. Shells were also used as currencies in the form of what people call cowries. And all over the Pacific, and not only Pacific, but also in Africa, there were currencies in the form of shells. Another use, which is very interesting, the first use of glue, the glue coming from bone and skin collagen. So if you boil skin or if you boil bones, you will extract, of course, some collagen. You will degrade collagen. And do you know what collagen is, by the way? What it is? What is it? Yes, it's a protein. I will come back later to that. So collagen has this special, even etymologically speaking, collagen to generate glue. Because people in the past understood that when you degrade collagen by heat, you will obtain this jelly, this glue, which acts as glue. So you had the first glue already 2,000 or 3,000 years ago. Another very interesting example of the use of biominerals, it's by the Maya. It was about 800 years ago. If you see on this jaw, you find that it's a normal jaw, but not completely normal, because or maybe I can just, if you look at, okay, I will. Okay, no, don't worry. I will just describe it. Okay, thank you, Mao. So if you look at this jaw, you feel that it's a sort of normal jaw, but if you look here, and here, these teeth are made of nacre, of mother of pearl. So meaning that 800 years ago, the Maya had the ability to replace missing teeth by nacre implants. And apparently the implants were completely tolerated by the bone or the jaw bones. So that's probably the first example of the use of nacre as a substitution material for bone or teeth repair. And as I said, all these teeth were completely osteo-integrated without any rejection. So that's very fascinating. So if you look in the Middle Age, for example, some other examples, in India, you had these shells with little Buddha. So all kinds of religious symbols were associated with shells, so with biominerals. So here, little Buddhas, and you have here the pecten, the scallop, which was used by pilgrims who went to Santiago de Compostela. After the discovery of the New World, in many churches, you had these fonts made of shells. So after the 16th century, the discovery of Asia and so on. And of course, lots of marquetry was made with nacre here. You had also a lot of manufactured objects. I show here this Nautilus shell, which is inserted into silver, into metal, and making these very beautiful objects. Very easily, you have these buttons made of nacre. Of course, you have jewels like pearls, different types of pearls. And also, I was mentioning the first use of glue, of collagen, extraction of collagen for making glue. Keep in mind that in the 17th century, in Holland, you had the first factory who made glue from collagen. The first industrial production of collagen that was in Holland, and probably for making this glue for probably these wallpapers, something like that. A lot of biominerals were used, or were and still are used for soil enrichment. And now let's go to the more scientific aspects of biomineralization as a field. We can go as far as Vesalius with what people call the basal skeleton. Vesalius was the first to describe very precisely the skeleton, human skeleton, which is kept in the Anatomical Museum of the Basel University. And a little bit later, you had, again, two Dutch people, Jan Swammerdam and Anton van Leeuwenhoek, were probably the first to invent the very, very first microscope. And this had of course, had its importance in the fact that very soon after that, people started to look at objects, among which biominerals. And for example, if you look in the well-known book of Robert Hooke, who was an English scientist, naturalist, he wrote this book, Micrographia. And in his book, there is one page devoted to, he looked at urine under the microscope, and he saw that in urine you have little gravels, little biominerals, of course, pathological biominerals, but he was the first to describe that actually in 1665. A bit later, another English guy, Clopton Havers, who made the first description of bone microstructure in his well-known monography called Osteologianova, or some new observation on bone and the parts belonging to them with the manner of their accretion and nutrition. So that was 1681. Then you had a French naturalist called de Lasson, who made the first experiment of burning bone and to see what was the composition of the chemical composition of the bone. Let's jump to the 19th century. Two French chemists, Racodeau on one side and Audier, were the first to to describe and discover chitin. Sounds a bit not related to the field, but actually we will see together that chitin is very important molecule in biomineralization. By the way, do you know what is chitin from a chemical viewpoint? Do you have any idea what is chitin? No, it's not a protein. It sounds like a protein, but it's not a protein. Now, actually, chitin is a very complex sugar. Chitin is a polymer of N-acetylglucosamine. So when you polymerize N-acetylglucosamine, you have this long chain and you make chitin. Okay, so that's quite important. Then 2 or 3 decades later, there was another guy called Frédéric, who was a chemist, a French chemist, who gave the first description, chemical characterization of different biominerals. And that's very interesting because he made these monographs of about 100 pages in Annales de Sciences Naturelles or something like that. That's in French. And he describes the chemical composition of bone, of teeth, of crustacean teguments, but also of Gorgonian exoskeletons, and even of mollusk shell. And for the little history, for the mollusk shell, he was the first to introduce the terminology nacreime, so which has been used much after, but he was the first to invented the term to describe this sort of proteinaceous component that you can extract from nature, from mother of pearl. So tell me if you don't understand, or if you just raise your hand and don't hesitate to stop me if I'm too fast or not very clear. Okay. So going further, if we go to the second half of the 19th century and first half of the 20th century, there were many descriptions of biominerals from optical microscope, and they are the contribution of 2 guys, a German guy, Schmidt, in the 1920s, who did some extraordinary work on that, and also a guy called Bogill, who was Danish or Swedish, I don't remember exactly, but who was the first to very precisely describe the structure of shells, of mollusk shells. And in parallel, there were a lot of chemical analysis of biominerals. So let's jump to the after-war period. After-war period is marked by the development of a novel discipline called biochemistry. And biochemistry was especially characterized by the development of new techniques for fractionation of molecules, of biomolecules. And at that time, when I speak at that time, I mean in the 50s, the main way to analyze proteins was to make these hydrolysis with acid. So you completely cleave in amino acids the proteins, and then you analyze in bulk the amino acids. That was the way people were doing it. No sequence, not yet any sequence, but still only amino acid composition of proteins. And if you look in the mid-50s, in parallel, there were people publishing amino acid composition of bone, of teeth, and shells. So all these publications came more or less at the same period, between 1953, 54, 55. In parallel, physical approaches included the X-ray diffraction of biomolecules. And for example, somebody like Ramachandran, who was a well-known Indian physiologist or physicist, he was the first to publish the 3D structure of collagen. And do you have any idea of what is the structure of collagen, by the way, the 3D structure of collagen? Yes? I don't, okay. No, it's a triple helix. So you have the double DNA helix, but you have also the triple helix of collagen. And this structure was really shown by this guy, Ramachandran, in 1954. In the 60s, of course, still a lot of biochemical characterization of biominerals. You had also in parallel the development of electron microscopy, TEM, but also SEM. And in the 70s, it has to be noted that there was the first international symposium on biomineralization, and that was in Mainz in 1970. Now we are already at the 17th symposium of biomineralization. It means that every 2 or 3 years, it depends, there is no complete periodicity of this for this event, but there is really a symposium that is being continued. Also in the 70s, we note the first molecular models on biomineralization. And I would like to give few names that are very important in the field. You have, for example, the name of Heinz Lowenstein, who was really one of the founding fathers of biomineralization as a discipline. And Heinz Lowenstein was probably the first scientist who made a very long list of all biominerals found on Earth. From bacteria to human beings, and he made this very extensive list of, and he published this paper together with Steve Weiner on biomineralization in 1989, which is really a sort of Bible of biomineralization. And in parallel, there was another book published by Kenneth Simkis and Carl Wilbur the same year, which was also dealing with biomineralization, but more from a cellular viewpoint. But having these two books is really the basis for biomineralization. So what about today? Today, as I said, there is still this international symposium of biomineralization, but there are also several important events like the Gordon Research Conference on Biomineralization, which is held every 2 years in summer. The ICCBMT, the International Conference on the Chemistry and Biology of Mineralized Tissue, which is every 3 years also. And we have also a lot of international or national conferences with a specific session devoted to biomineralization. So that's what we can say for the discipline. Now I have sort of finished with the purely historical aspect of biomineralization, and we are going to look a little bit together what I'm speaking about. So biomineralization is a very widespread phenomenon because it goes from bacteria till vertebrates, until elephant. And it concerns about 55 phyla, living or fossil, among which phyla in the bacterial world, in the archaeal world, but also in the eukaryotic cells world. And if you look at the different types of mineral produced by living system, it's about 70 different minerals. So it's not a lot. If you compare if you look at the minerals that are made by nature, but in a completely abiotic way, you have more than 3,000 minerals. But for the biomineralization world, it's more about 70 minerals. And these biominerals are, let's say, between different type of chemistry. For example, here you find iron oxides, very common in the bacterial world. Manganese oxides, also that are characteristic of the bacterial world. Sulfides, oxalates. Oxalates are some plants can make oxalates, but we also make oxalates. Do you know where? Do you know where we make oxalates? Do you have any idea? Well, if you have problems with your kidneys, if you have kidney stones, many in 70% of the time they are made of oxalates, calcium oxalates. You also have sulfates. Sulfates are very rare minerals, I would say. You find them, for example, in some jellyfishes as a statolix, so a structure which allows the jellyfishes to stabilize in the water. You have also organic minerals. Aha, what are organic minerals? Well, I can tell you these organic minerals are usually anhydrous uric acid. Anhydrous uric acid. These are also pathological mineralization in kidneys. So about 20 or 30% of the cases, they are anhydrous uric acid. You have halides, very, very rare. You find them in some gizzard plates of some gastropods. It's calcium fluoride, but it's very anecdotic. Of course, phosphates. Phosphates are the teeth and the bones. You have silica. Silica, for example, in diatoms or radiolarians, and carbonates. And I will mostly focus the second part of my talk to carbonates. Well, it is already known for 30 years or a bit more than people tend to split the different type of biomineralization in two big categories. One is called the biologically induced mineralization, and the second category is the biologically controlled mineralization. So we are going to see together what the differences between these two types of biomineralization. Let's start first with the biologically induced mineralization. Well, they are supposed not to require very, very specific macromolecular machinery. Of course, this is a little bit tricky to say that, but it seems that there is some truth in it. The formed crystals tend to look like chemically precipitated crystals. The biologically induced mineralization are rather strongly dependent on environmental conditions, meaning that if the conditions are not favorable for making biominerals, then the organism will not produce these biominerals. And of course, there is no control on the shape and layout of the crystals. So which kind of organisms are concerned by these type of biologically induced mineralization? Well, many different bacteria, some fungi, some protists, some algae, and also you have this pathological mineralization in metazoans. One well-known example of biologically induced mineralization is provided by stromatolites. So have you heard already about stromatolites? No? Never? The stromatolites are organo-sedimentary structure made by actually the activity of a bacterial community, in particular cyanobacterial communities. And they made this structure, I'm going to show later, which can be centimetric, decimetric, or metric, and they can produce really huge amount of material of calcium carbonate. And they are usually concentric structure, like shown in the picture. You have this type of material that you can find in which are recently formed, like here in Turkey, but you have the same in the Bahamas. And if you look the communities which make this type of structure, usually you find a mixture of different communities, like we have the phototrophic bacteria, which are the cyanobacteria, which are the main players. But you find sometimes also heterotrophic and aerobic bacteria, and more inside the structure, you find these heterotrophic anaerobic bacteria. And these communities are able to really make laminations, like you can see here, which can be really of big size. And some of them are really fossil material, but in the living system, you also find these stromatolites. Of course, the most ancient stromatolites are 3.5 billion years old, and they are found in Australia and also South Africa. And I will come back later to this when we look at the evolution of the biomineralization. but one has to know that the same structure, exactly the same, are also present in living system. And the best place to see that is Shark Bay in Australia, where you find these stromatolites. Again, these structure, which are mostly made in the sea. And let's go to the second type of biomineralization, that is the biologically controlled mineralization. So here again, you have different criteria which help to understand what these biominizations are. First of all, this biomineralization, or this type of biomineralization, requires a very specific molecular and cellular machinery, and I will explain that. You have also the problem of the space delineation. Usually, the crystallization in biologically controlled mineralization is not in open access to the sea. It's made in a closed, confined, closed environment, and we will see that too. The form crystals are very different from their chemical counterparts. You have also a multi-scale organization of this type of biomineralization. These biominizations are also far less dependent on environmental conditions, which means that even if the conditions are not favorable to make this type of biomineralization, the animal still can do it. I will show some examples. And of course, the last criterion is that mineral deposition is really controlled by what we call an organic matrix. And we will see that too. So which kind of organisms are concerned by this type of biologically controlled mineralization? You have, for example, in the bacterial world, you have the magnetotactic bacteria, but you have also many protist organisms, so single-cell eukaryotes. You have some algae, in the broad sense, and of course, many metazoans. When I speak about metazoans, is it clear for you, metazoans? What could be a synonym of metazoans? Animals. So metazoans are pluricellular animals. So we are metazoans. Huh? Okay. So let's detail a little bit each of these criteria. The first criteria, very specific molecular cellular machinery. If you look, for example, in the type for the bone tissue, you have really the triple row of 3 different cell types, which are the osteoblast, you have the osteocytes, the osteoclast. So the osteoblasts are the ones which produce collagen, bone collagen. Osteoclasts are the ones which degrade bone collagen. So you have already different cells. But the cells are not isolated, and usually they are made in a special organ. If you take the example of the mollusk makes its shell owing to one specific organ, which is the mantle. And the mantle is really what you see here. Here you have a pearl oyster which has been opened in two, and the mantle is really the organ which spreads on the shell, and this is the one which makes the shell. Of course, this organ appears very early during development by cell differentiation, and upstream the formation of these organs, you have a complete what people call a gene regulatory network, which, for example, in the case of sea urchin, has been very well studied. So it means that there is a complete machinery. That's that's the main message I want to share with you. Complete machinery to make the biominerals. Again, for this aspect, I show here more precisely the example of the edible Pacific oyster, Crassostrea gigas, which is now called Magallana gigas. And you see, so are there people here who eat oysters during Christmas time? Do you eat, you don't eat oysters? You should. That's my feeling. Well, I eat a lot of oysters during Christmas time. I'm a big fan. So I just open them. When you open the oyster, you see this organ. Huh? This organ is really the mantle. And the mantle is really the organ which makes the shell. So in oyster, the mantle is very thin and almost transparent. And when you see the thickness of the shell, you say, wow, it's possible that such a thin organ can produce so much calcium carbonate, but it does. And actually, there is a complex chemistry happening, taking place in the mantle, because you need calcium bicarbonate, which has to react with, you have bicarbonate, which reacts with calcium ions, which is supposed to make calcium carbonate and also release one proton, and the proton is has to be translocated. So what happens actually is that all the ingredients for making calcium carbonate are concentrated here in the mantle tissue. Sometimes they are concentrated in the form of amorphous granules, not always, but, and in the meantime, you have the shell matrix secretion by the same tissue, and the protons are reabsorbed. And this is a very, very simplified view. We will see later that it's more complex than that. But what I mean, again, here, that you need a very complete machinery to make a shell. The second criterion, space delineation, which means that in biologically controlled mineralization, minerals are never produced in direct contact with the ambient medium. And there is a very nice example. These examples are the coccolithophore algae. So do you know coccolithophore algae? Does it sound, yes? Everybody? Okay. So coccolithophore algae are small algae. They are single-cell algae. And they make these little plates that are called coccoliths, usually in calcite, sometimes in aragonite, but most of the time, at least for this animal, which is Emiliania huxleyi, one of the most well-known coccolithophore algae, it's calcite. And there was a description of this, the making of each coccolith. Actually, what happens is that you have the single cell here with the nucleus, a big chloroplast sticked with the nucleus, and also here, a sort of vesicle which is fed by the Golgi apparatus. And while it is fed, you have the production of a sort of basal plate here, which will grow, and the basal plate will progressively calcify. And in this vesicle, so it's really a confined space with specific properties, and then later the vesicle detached from the nucleus and will migrate to the outer border of the cell. This is how coccoliths are formed. Again, a very sort of rapid view. But what I mean here is that, again, here you have each coccolith is formed in a very confined space. Another example of the space delineation or space confinement is, again, the mollusk shell. Because the shell itself is not made in direct contact with the seawater or with the fresh water, it depends. But the animal, before producing a shell, it will produce what we call a periostracum, which is actually, everybody knows the periostracum. Do you eat mussels, edible mussels? Yeah, okay. Mulfrit is good. So if you look at the outer surface of mussels, it's a little bit blackish, and it's a bit leathery. And this leathery part that you can even pull out of the shell is really the periostracum. So it's very well developed in mussels. In some other bivalves, it's less developed, but still, this periostracum layer is the one that seals the space between the mantle tissue here and the site where the shell grows. Actually, the periostracum is really the sealing layer which makes the shell made in a confined space. The third criterion is about the fact that the form crystals in biologically controlled mineralization are very different from their chemical counterpart. And there is a very nice example. Here you have a single crystal of calcite, which is made very completely abiotically. So it's very easy because you produce, you let diffuse ammonium bicarbonate in a solution of calcium chloride. And then if you do that for a few days, you can end up with this type of very nice rhombohedron, which are characteristic of calcite. Okay. Now, if you look here, sea urchin spines. So if you take one of these sea urchin spine, and if you do some X-ray diffraction on one spine, you will see that one spine behaves like a single crystal. So if you do diffraction here and diffraction here, you will see one pattern of diffraction, and they will diffract all together like single crystals. So from a let's say crystallographic perspectives, the abiotic calcite and biological calcite are just similar. But of course, now if you look under the SEM, a spine, a sea urchin spine, you see that it's very complicated. So you see one of these, it's here, and then you focus again, you see that you see some substructure, which are, so at different scales, you see that the sea urchin spine is very complicated, but still it looks like a single crystal. So again, the form crystals are very, very different from their equivalent counterpart, from their chemical counterpart. Another criterion which is important is that you have this multi-scale organization. And the typical example is that of bone, huh? Because we are speaking about collagen. So collagen, you have this, the monomer of collagen is a single chain, but this single chain will associate with two other chains to make this triple helix, okay? So we have already two levels of organization. This triple helix will associate in parallel and to form this structure, which can make these mineralized fibers. These mineralized fibers will associate in fiber bundles, which will associate in forming lamella, and lamella will make osteon. So you have 7 or 8 different levels of organization. So the bone is a very complex tissue. No? We could say maybe it's because of vertebrates. Vertebrates are a bit different from the other. But if you look, the red color, and it's also a very interesting example because red color, corallium rubrum, is made of calcite, which is a little bit enriched in magnesium. And red coral has been very well studied by one crystallograph in Marseille, Daniel Vielzeuf, some years ago. He published a series of papers where he showed that, again, for the red coral, you have also this hierarchical organization of the skeleton. So when you take a branch and cut it, you will see this sort of structure. Each of these structures is composed of what Daniel Vielzeuf called 2 herringbone units, which are themselves made of spindles. The spindles are made of little lozenges here, which are themselves made of very fine fibers, less than about 1 micron in thickness. And then these fibers are also made of little crystals, which are themselves made of nanodomains. So you have a very, very complex organization. So even for, let's say, between brackets, simple animal, red coral is not simple, but it's already very complex. But for this type of animals, you have also this structure, multi-layer, multi-scale organization. The fifth criterion is about the fact that biologically controlled mineralization are far less dependent on environmental conditions. And there is these very beautiful examples of acantharians. Acantharians are protists, which are planktonic. They are marine animals, they are planktonic. And they made these little tests, these little biomineralization made of strontium sulfate. Strontium sulfate is a very rare mineral. And it's called celestite. And strontium sulfate is very undersaturated in marine environment. It means that if you put a strontium sulfate crystal in seawater, it will just dissolve. It's because of its undersaturation. But this animal makes these strontium sulfate biominerals, and nobody knows why. There is no reason for it. It means that a big part of its energetic budget is devoted to make constantly these strontium sulfate tests. Another example, two other examples, which are taken from metazoans. For example, if you go in the western part of Burgundy, you have, let's say, Morvan. Morvan is a granitic area. So the waters in Morvan, the fresh water are rather acidic, and they are very poor in calcium. But in this area, you can find these big shells with very thick layer of nacre, and these animals have this very low metabolism. They produce nacre very, very slowly, and they can live very long, like 80 years. And they still, in spite of the fact that the water are acidic and very poor in calcium, the animal is able to make a very thick and very big shell. The second example is the ascidian. Ascidians are urochordates, and one specific, one genus or one species is known, Erdmania momus is known for making these little spicules in the teguments, and these spicules are made of vaterite. And vaterite is a polymorph of calcium carbonate, which is very unstable. So normally vaterite does not exist like this in the living system. But this animal makes vaterites. And again, nobody knows why vaterite and not calcite or aragonite. So again, here, these examples are really to show that in biologically controlled mineralization, you can make minerals even if these minerals are not favored by the environment. And finally, biologically controlled mineralization are controlled by an organic matrix. And I show here a few examples of in different systems. For example, in diatoms, in the silica world, you have diatoms, and we know now that diatoms are a complete set of proteins that are supposed to control the deposition of silica. You have silafins, frustulins, and so on. You have also, in the demosponge world, you have also a lot of proteins like silicateins, galactins. You have also some collagens, selenoproteins, and so on, silicase, which are also associated to the skeletal matrix. In sea urchins, which produce high magnesium calcite, you have a complete set of different proteins like MSP 130, SM50, SM32, SM37, and so on. So you have a complete list of proteins associated to the spicules and skeleton of sea urchins. In mollusks also, we have a very long list. I just show here a few of them. And of course, in chordates, for example, for the teeth, I don't speak about the bones, but just for the teeth, you have a lot of different proteins like amylogenins, ameloblastins, enamelins, amylotins, and so on, so on. So all these proteins are associated to the minerals tissue. And in all of these examples, I also speak about PTMS. PTMS are post-translational modifications, because many of these proteins, so do you know what post-translational modifications are? So when you have a protein, so protein is, let's say, a chain of amino acids, okay? Chain of amino acids. And many of these amino acids will accept, some of these amino acids will accept some extra functions like sugars. So some proteins will be glycosylated, they will have sugars which are attached, or some of them will be phosphorylated, so they accept from phosphate groups and so on. So all these post-transactional modification, which is very common in the living systems, they are present in proteins associated to biominerals. Okay? So now I'm going to sort of screen some examples of biologically controlled mineralization. And the first examples are the magnetotactic bacteria. We don't know, actually, it's very bizarre to imagine that some bacteria have to orient in the magnetic field. That's the reality. And some of these magnetotactic bacteria, on all of them, they produce magnetite nanograins from 25 to 100 nanometers, which are synthesized in a special organite, in a special sort of vesicle, which is called a magnetosome. And all this process is controlled by about 20 different proteins. It has been very well described, especially by Dirk Schuller, a German scientist who really has completely described the equipment for making magnetite particles. Another very interesting example, and that was published by a French group in Paris, the group of Karim Benzerara. They found photosynthetic bacteria, so cyanobacteria, forming intracellular calcium nodules. And this process is completely controlled. It's not just a secretion of calcium carbonate. No, it's fully controlled. They know the genetic of that. And of course, we don't know exactly what could be the function of these intracellular granules. Some people, they say that it might work as a ballast or something for making the cell a bit more heavy. So I don't know. But at least this process is very controlled. Of course, we have this example of the coccolithophore. I will not really insist on that, but it's a very controlled process. It has been very well described by different groups, especially in Holland, but also in England. Another example are the diatoms. So the diatoms are, and they use silica, and they have colonized all the environment, all the aqueous environment from seawater, but also fresh water. And they are really key players nowadays in the regulation of silica at the global scale. Another examples are the foraminifera. So the foraminifera, they are single-cell organisms, so protozoans, that are able to make either calcite or aragonite test or skeleton. And there are well-known examples in the past. These are the nummulids, which were really cells which produce a huge amount of calcium carbonate about 40 to 50 million years ago. But nowadays, you have foraminifera, which are also present in as benthic organisms or as planktonic organisms. Another interesting example, if we go now to the metazoan world, to the animal world, to the pluricellular animal world, this is the example of sponges, because sponges, it's sort of complex group, like between 5 and 6,000 living species. Some of them are able to make silica, like this animal, and it's called a glass sponge, and they are called hexactinellid sponges. So they use silica and they make it a sort of very precise skeleton of silica. Some sponges are able to make calcareous spicules, like here. And you even have some sponges which are able to make a calcareous exoskeleton together with some silica spicules. There is a little tiny group of demo sponges which are able to manipulate the two different minerals. So it's very fascinating. Well, another well-known example are that of cnidarian, and of course, cnidarian, you know the corals, which make these very beautiful reefs everywhere, especially in tropical areas. So this is a picture from French Polynesia, where you have these 140 different islands spread on a territory which has the size of Europe. And if you look from a plane, you see these little rings. It's sort of fascinating because all these calcium carbonate. And in the cnidarians, you have let's say the reef building corals, and they called scleractinian corals, and they use aragonite. So that's one form of calcium carbonate. And beside, you have some octocorals, like the red coral. They use calcite, so another polymorph of calcium carbonate. So do you know what a polymorph is in crystallography? So two polymorphs have the same chemical formula, like calcium carbonate, CaCO3, but it's they crystallize in different systems. So in the case of calcite, it crystallizes as a rhombohedron in a rhombohedric system, while aragonite crystallizes in orthorhombic system. So it's a little bit different. But the formula, the chemical formula is exactly the same. Other examples, which are a little bit less known, that of brachiopods and bryozoans, and they mostly use calcite and a little bit of aragonite in the case of some bryozoans. And the case of brachiopods is interesting. Brachiopods look like mollusks, but they are not at all mollusks. They have a bivalve shell, but they are completely different from a mollusk, from their structure. But they use calcite for making their shell. There is one exception, some brachiopods that use calcium phosphate, and they are sort of very close to what the human bone system. Another example, which is also very interesting, is that of arthropods, of crustaceans. When I speak about crustaceans, I speak about lobsters, crayfishes, shrimp, prawn, crabs, because they have these cuticle, this exoskeleton, and this exoskeleton has to be renewed every time because the animal has to mold to grow in size. So it means that this system is not a system which grows during all the life of the animal, but the animal has to mold from time to time, so to change its suit and then to find another one. And this cuticle is usually mineralized, so it can be very heavily mineralized for lobsters and crabs, and sometimes less mineralized for prawns. But what is interesting is that together with this external skeletal tissue, you have also these what people call gastrolytes. And for example, if you dissect a crayfish, if you remove the this part, you will find this type of structure, which are at the basis of the cephalon, or the head. And these structures are calcium carbonate. They are like little bits of calcium carbonates, and they are storage structure. So it means that when the animal needs calcium carbonate, it will slowly dissolve this structure to be able to mineralize the new cuticle. So it means that in this case of crustaceans, the biomineralization is completely dependent on the molting hormone. So it's really on you have a signal, an hormone, which gives the animal the order to remove its cuticle. So this hormone is really controlling the whole biomineralization process. One example which has been studied by a former colleague in Dijon is that of a very tiny terrestrial amphipod crustaceans called Orchestia calimana. Orchestia looks like a little shrimp, very tiny, less than 1 centimeter in length. And if you make X-ray from this animal, you will see that inside the animal, there are like 2 tubes which are opaque to X-ray. And these 2 tubes are actually 2 concretions, like that you can see here. And these 2 concretions are made of calcium carbonate of a special kind. It's not calcite, it's not aragonite, it's not vaterite, it's amorphous calcium carbonate. And why amorphous calcium carbonate? Do you have any idea? I will tell you. Amorphous calcium carbonate is easier to dissolve. If you have a crystallized form of calcium carbonate, you need a certain energy to dissolve it. But when it's amorphous, so when it's less structured than the crystallized form, then you can dissolve it more easily. And you can imagine that these 2 little tubes here, so when the animal molts, so remove its cuticle, it needs to get, so it has its new cuticle, which is very soft at the beginning, and it has to be mineralized very quickly. So what the animal does is to dissolve partly the cuticle, and you see here the cuticle, the concretion before the molting process, and after the molting process, you see that it has been partly dissolved. So this part, which is partly dissolved, then will migrate to harden the cuticle. So you have a very elegant system here where you have these calcium storage structure, which is involved in the mineralization of the cuticle. Well, another examples are the mollusks, and I will tell more in detail what it is about. Mollusks are very interesting because they can produce these very huge accumulation of shells, what people call thanatocyanosis. And in the past, and I will come back later to this aspect, some mollusks were able to make complete reefs, huge reefs, especially about 80 to 90 million years ago. And finally, you have the vertebrate with the bone and the teeth. So I have finished the first, the introduction of my talk. So I don't know what time it is. I can directly start with the second one, or do you want to get a little... 3:45, so I have spoken for 6:30. Okay, so I can continue with the second one if you want.
Yeah.
Okay. Yeah, is it okay? You are not yet dead? So don't hesitate to ask questions if necessary. How can I get the second one?
Okay. This is right.
Super, merci. So now we are going to go a little bit more in detail about the origin of evolution of marine biocalcification. No? So it will be, I think it will be a rather long, probably more than 1 hour. Then I will stop after that. We will have a little pause and then we will continue the others. So again, I show here the different minerals that are produced by living systems. And we will refocus on what is probably the most important from a quantitative viewpoint at the surface of the Earth. These are carbonates. So first of all, carbonates are a bit everywhere. And I would like to start with few remarks. If you look at the seawater is highly supersaturated with respect to calcium carbonate. However, so it means that normally calcium carbonate should precipitate everywhere massively in seawater. It does not occur. It's not the case. So a place where spontaneous calcium carbonate precipitation occur are very rare, like in the Bahamas, for example. You may have some places where you have really spontaneous precipitation of calcium carbonate. But in most of the places, this does not happen. Why? It's because in the seawater, you have a lot of inhibitors of precipitation. So seawater is very saturated with respect to calcium carbonate, but the calcium carbonate does not precipitate. Why? Because you have magnesium, for example. You have a lot of organic polymers floating everywhere, polysaccharide proteins, and you have also phosphates, citrates, and so on. So a lot of polymer, mainly organic polymers, which are just preventing the calcification, the production of calcium carbonate in seawater. Now, if you look from a global scale, calcium carbonate represent about, or it's said to represent about 4% of the Earth's crust. It's not a lot. One says that the total production of calcium carbonate per year should be between 9 and 14 or 15 billion tons, so per year. And in this huge production, you have the marine biogenic calcium carbonate, which represent maybe 1/2 or maybe even more. So it's from 5 to 6 billion tons a year. And the production of calcium carbonate, actually, you can see it very easily. So I was referring to the reef system here, which are really living systems where you have this intensive production of calcium carbonate. But also, if you look in the past, you have these cliffs, these limestone cliffs, for example, in Normandy or in Brittany. You have also these huge cliffs made of calcium carbonate in the south of France, but also in Italy, everywhere. So this production of calcium carbonate is very important. So if you now focus on the production of marine calcium carbonate, biogenic calcium carbonate, I was saying that it's between 5 and 6 billion tons a year. A part of this production will be redissolved rather quickly. And do you know why it will be redissolved? Yes?
Yeah. Can you repeat?
Yeah. So the yearly production of calcium carbonate is about between 5 and 6 billion tons a year. And from this large amount, about 40%, that's an estimation, about 40% will be redissolved. Do you know why? Actually, it has to do with the compensation line of carbonates. So when carbonates are sinking in the seawater, below a certain depth, they will just redissolve. And this happens especially in the open oceans. So a part of the system will, a part of the calcium carbonate will be sort of re-injected as calcium ions and bicarbonate or carbonate. But 60% of this calcium carbonate, they will just accumulate, making layers. And these numbers, 5 to 6 billion tons a year, are really for the marine systems, meaning what we call the continental shelf here, so the epicontinental seawater, and the open oceans. Okay. Now, if we try to make a sort of list of what happens in the different systems, so we have again the continental shelf here, epicontinental platform, and the production is estimated to be around 2.5 billion tons a year. The coral themselves, they are able to make 1/3 of this production, which is quite a lot. And this production is mainly benthic, so it's really at the bottom of the seawater. It's mostly aragonite and calcite or magnesium calcite. And the main producers are corals, foraminifera, mollusks, bryozoans, red algae, green algae. So a complete list of organisms that are able to make calcium carbonate in benthic systems. And you have also the open oceans. The open oceans are, of course, very much deeper. And the production of the open oceans, it's more or less the same number as here. It's 2.4 billion tons a year. And this production is mainly planktonic. And it's made a lot of calcite. And the main organism which produce these planktonic calcium carbonate are coccolithophore algae. But you have also some planktonic foraminifera, which also produce aragonite. And you have also a group of specific gasteropods called pteropods. Have you heard already about pteropods? Yes? No? Pteropods are very specific. They are tiny gastropods, so tiny mollusks, which are planktonic. So they are a couple of milliliters in size, or sometimes less. And they are really planktonic animals. And they were a little bit overshadowed by coccolithophore algae for a long time. But now people start to see that pteropods are maybe important in the global production of calcium carbonate at the surface of the Earth, especially in open oceans. So in these two systems, the things are quite well established. What happens on the continents? Well, actually, production of calcium carbonate on the continents is completely unknown for different reasons, because it's very patchy. There are some huge domains like deserts, like hot deserts, where the production of calcium carbonate is very limited. You may have tiny gastropods that can produce a bit of calcium carbonate in deserts. But in temperate forests, you have a lot of microorganisms like bacteria, but also fungi, which can produce a lot of calcium carbonate, especially the calcite in needles. And in tropical areas, the production of calcium carbonate can be very important. And there is a very beautiful example, the example of the Iroko trees. So these trees are massive trees, and they produce in their roots, around their roots, a huge amount of calcium carbonate, owing to some bacteria associating with the roots. So there is a complete, it has been well documented, this system, where these oxalotrophic bacteria contribute to the production of calcium carbonate at the basis of the roots of the tree. So one tree can produce like 10 tons of calcium carbonate, which is quite enormous. So locally, you can have a huge production of calcium carbonate, but this is only locally. So
the wet ring, is that
Yeah, also, yeah. Yeah, you're right. Yeah. I don't speak about that especially in the continents, but of course, yeah, the weathering can really contribute a lot to production of. So it can also in the lacustrine system in lakes, you can have also a huge production of calcium carbonates. For example, in Jura, in France, maybe 100 kilometers from Dijon, we have some lakes where the production of calcium carbonate is really huge, but that's really local. So the question is, was this calcifying regime constant across the geological time? And the answer is, of course, no. And we are going to see together a little bit about the evolution of this production of calcium carbonate. I just start with the macroevolution of calcium carbonate-based biomineralization across geological times, and I just show here this geological time scale, the full scale, which goes from the beginning of the Earth, 4.6 billion years ago, until now. And you can see here, I just represent at the right scale the history of the Earth. So the very long tail called the Phanerozoic, that goes from the Cambrian until now, is actually 1/7 or 1/8 of the whole history of the Earth. And most of the history of the Earth is Precambrian, which is itself divided in Hadean, Archean, and Proterozoic. So the Hadean is really the period where the Earth was formed, and the prebiotic Earth without life. So it goes from 4.6 to about 3.8 billion years ago. Then you have the Archean, where you have the development of early forms of life from 3.8 to 2.5. Then the Proterozoic from 2.5 until 0.54 billion years. So we are going to sort of sweep across all these periods. And first, what happens during the Hadean times? So one first remark is that calcium carbonate and calcium carbonate, they were not formed at during formation of the Earth. They came very secondarily. Why? Imagine that the Earth was, I will not speak in detail about the formation of the Earth. Actually, I was not there, so, but what can be said is that when the Earth started its accretion, all the heavy elements like iron, nickel, they migrated more in the center, and the lighter elements or the lighter form the silicates, and the silicates form the crust. And when the crust was formed, what happened was that you had release of CO2, release of water. You have also bombing by meteorites, so meteorites which could also bring water. So you had the formation of these proto-oceans. So proto-oceans, so water plus CO2, which could go into the water, which could be dissolved. These were the ingredients that sort of contributed to the weathering of silicates. And the weathering of silicates released calcium, and the free calcium could very spontaneously precipitate together with CO2 or bicarbonate. So you probably, the first calcium carbonate that were formed was probably during the Hadean time, but not at the beginning, not at the very beginning, but somewhere later, where you could have this first precipitation of calcium carbonate, spontaneous precipitation of calcium carbonate. Of course, very soon, this system could be amplified by the fact that life appeared, especially cyanobacteria which could make these stromatolites. So another idea which is interesting to think about is that during the Hadean time, so we had these proto-oceans, you had also a lot of polymer inside, a lot of organic polymers. So we can even think that during that time, you could have a sort of selections of polymer at the surface of minerals, and some polymers were probably selected because they had a strong affinity for mineral surfaces, and some other polymers were probably recycled and not really selected by mineral surfaces. So you had maybe the first premise, or I don't know how to say that, the first way of interaction between a mineral phase and some organics. So it's not yet biomineralization, of course, but it's something that people can call organomineralization. So that was during the Hadean time. But of course, we don't have any clue, anything from this period and from what happened during that period. So if we go to the Archean Proterozoic times, then we see the first biologically induced mineralization and probably also the appearance of the first biologically controlled mineralization. So I was referring to the stromatolites before. So as I said, these are the oldest mineralization construction since they started to appear about 3.5 billion years ago in the Archean times. Again, you see they are living equivalent in the Shark Bay in Australia, and they are exactly the same as probably they were in the Archean times, so the same way of functioning. And of course, the stromatolites are really the examples where you probably had a coupling between the photosynthesis and biomineralization. And as you know, the photosynthesis means that you use CO2, carbon dioxide, and you transform it into organic matter and oxygen. So, of course, stromatolites are supposed to have played a key role in the formation of an oxidizing atmosphere. Well, if we look more in detail, we see that people speak about the GOE, so the Great Oxidation Event, which started in the Paleoproterozoic, where so during the Archean times, the level of oxygen was still very low, but then probably it increased a little bit in the Proterozoic here, and that's during this period. And then it reached a sort of plateau during the Mesoproterozoic and part of the Neoproterozoic before growing again. So these are the let's say the most recent findings that people made about the evolution of the atmospheric oxygen on the Earth. And of course, having this oxygen level rising, probably contributed to the extinction of a lot of anaerobic life at the surface of the Earth. So the stromatolites, as I said, they are organomineral constructions made by cyanobacteria community, but not only, some other bacteria. But probably there was an advantage in having stromatolites because it makes a support. It can also structure bacteria communities. It's somehow protection against desiccation. And for some lower bacteria which are in the lower part of the stromatolites, they also sort of give the protections against UV. So there must have been some advantages of making stromatolites by cyanobacteria. And other things that are important to underline is that the production of these stromatolites are probably associated to the formation of what people call EPS, exopolymeric substances, which are actually amalgamates of sugars and proteins. And these EPS probably also contributed to the production or to the deposition of calcium carbonate in stromatolites. And again, here you may have had a peculiar microenvironment which could favor nucleation and templating the calcium carbonate, but also the system could probably work reversely. These EPS could also probably inhibit mineralization in some way. Of course, all these let's say rows of EPS are probably favored by the handling of intracellular calcium fluxes by these cyanobacterial cells. And you know that calcium has a special function in cells. It's called a second messenger. So probably very early, calcium was got this role for signaling, and very early in the history of life in the archaea, you had a complete cytoplasmic regulatory machinery which was set up even before the cells or bacterial cells started to calcify. Well, let's go closer. Let's go now to Proterozoic. So that's the last part of the Precambrian. And there are some traces of multicellular eukaryotic life, which are probably represented by fungi, and it's about 0.9 to 1 billion years ago. And it's interesting because we have here probably the first witness of the use of chitin as an ingredient. Of course, these animals were not mineralizing. They are really non-mineralizing organisms, but they already sort of invented chitin. And probably chitin was invented even earlier. So again, I will come back to chitin because it's an important ingredient in many mineralization systems. Let's go a little bit closer from Earth. You have already very complex biomineralized forms of life in some deposits in Yukon in Canada, which are represented by eukaryotic cells. And the mineral forms was hydroxyapatite, and the deposits that were studied, so that's less than 10 years ago, have been dated of about 810 million years ago. So we are again in the last part of the Precambrian, in the Proterozoic. And these are probably the first traces of biominerals. Now, let's go. Let's go more to this limit between the end of Proterozoic and the beginning of the Phanerozoic. We have this special event called the Cambrian explosion. So we are going to detail a little bit what happens there. And usually, this limit is really the limit from which biologically controlled mineralization started to be very dominant in living systems. So let's see what happens. Of course, as a preamble, biomineralization is one aspect of the Cambrian explosion. It's not the single one. I mean, the Cambrian explosion means it has been studied a lot by many people, and usually looking at biomineralization is one part of the Cambrian explosion, but not the only part. So if we sort of summarize what may have happened few tens of million years before the Cambrian explosion, we see, for example, the fossil embryos from Dushanto in China, and there is this Ediacara fauna. And the first mineralized form, not completely the first, but some, let's say, abundant mineralized form called Cludina, preceding by a few million years the limit between the Precambrian and the Cambrian. And then we have these small shelly fossils, and then the real Cambrian explosion. So we are going just to sort of travel from here to here. The Dushantuo formation in China is well known for the first fossil embryos, which are remarkably conserved, and they were published like 27 years ago. The hit of a couple of papers in Nature and Science and so on. And what is funny is that in this very old deposit, it's almost 600 million years old, you can find different stages of developmental formation, from egg till two-stage cells and morula and so on. So you have, and all this is made of calcium phosphate. So these very soft samples have been preserved as in calcium phosphate. And there is no, as far as I know, we don't know exactly which animal corresponded to these fossil embryos, either macroalgae, cnidarians, sponges, bilaterian, some simple adult metazoans. So different hypotheses have been formulated to try to relate these fossil embryos to living systems, to systems that are known. Very in parallel to these fossil embryos, which are very located especially in China, you have the development of a special fauna, which is called the Ediacara fauna, and which has been well studied. The Ediacara fauna has been discovered in 1947 in the Ediacara Hills in Australia. And after this initial discovery, in many places all around the world, the Ediacara fauna was discovered. And I just show here a few representatives of these fauna, like Dinkinsonia, Eoporpita, Tribrachidium, Kimberella. So Kimberella is, for example, supposed to be a sort of primitive mollusk without shell. So the Ediacara fauna was not mineralized. It was a soft-bodied fauna, which has a worldwide distribution, more than 30 sites on the five continents. It's a soft-bodied fauna, not mineralized. People think that it was mainly represented by animals which were macrophage, so which could eat small particles, let's say, which means that they were short trophic chains. This fauna is supposed to have been adapted to a low level of oxygen, rather low level in comparison to nowadays, because these organisms are flat, so which means that we had a high surface exchange with the seawater. And people who studied this fauna could really see that there is an increase of this fauna from 570 million years to 545 million years, and then this fauna disappeared. And people speak about 70 different genera reparted in about 100 species, but what is a species for a fauna which has disappeared? It's really a question. But a lot of this fauna is represented by cnidarians, so medusoid shapes animals, but also flatworms, arthropods, and also unknown phylum. What is interesting is that this fauna seems to have emerged after deglaciation. There was a glaciation called glacial glaciation, and this fauna seems to be more or less the development of this fauna seems to have been more or less associated to the deglaciation. So there are a few papers speaking about the late Neoproterozoic deep ocean oxygenation on the rise of animal life. So it's possible that the deglaciation induced the ocean oxygenation, which may have allowed the formation of this life. So, as I said, this fauna was discovered already a long time ago in different places. So it's a world, it has a world distribution, but still its connection with living life is questionable. Huh? Some people say that Ediacaran fauna has nothing to do with what we know from the metazoans. So they even speak about a group called Vandobionts, which would associate all these fauna. It's an aborted experiment without offspring. And but if we look more carefully, the Ediacaran fauna is also a mixture of representative of disappeared and living phylum, because one can recognize in this fauna some sponges, cnidarians, flatworms, arthropods, and maybe some echinoderms. And there is the case of Dickinsonia. So a few years ago, there was this interesting paper showing that in Dickinsonia, there were some lipidic biomarkers which with cholesteroid that would be typical of metazoans. So there was, I mean, this paper just showed that there may be some relationship between the Ediacaran fauna and what we know today and what emerged in the Cambrian time. So more or less in parallel to Ediacaran fauna, there was also the early mineralized forms that are just below the limit between Precambrian and Cambrian. I have to so. So these forms, they were let's say discovered the first time in 1972, were published, and it's called Cludina. So Cludina are really like tiny little tubes made of conical shape in each other, and they are supposed to be associated to be like cnidarians, and they have a world distribution. They are calcified, so made of calcium carbonate, lightly calcified, and they are about the same age as the Ediacaran fauna, and they also disappeared at the Precambrian-Cambrian transition. And since the discovery of Cludina, many other, or not many, but some other genera were discovered, like Namacalatus, Namapoikia, Cynotubulidés, which are more or less associated to these cludinid animals. What is interesting is that there are a couple of papers showing that these cludinids had some marks of predation, which led some people saying that maybe the first traces of predation came from that period. Before, there was no predation, and then predation appeared a little bit before the Precambrian-Cambrian limit. Now we can jump really above the limit between Precambrian to Cambrian, so about 542, 543 million years ago. And at the basis of the Cambrian, one finds the small shelly fossils. And the small shelly fossils, the SSF fauna, they have a world distribution. They are made of conical shapes, plates, clerite, tubes, And they represent probably the first diversification of skeleton-bearing organisms. About 40 genera have been described. Some of them have certain uncertain affinities. Some of them are associated to sponges, cnidarian, maybe very some mollusks. More or less at the same time, during the middle to lower Cambrian, in parallel to the small shelly fossils, some specific organisms appeared, which are called the archaeocyates. Have you heard about that already? Archaeocyates. Archaeocyates. No? Archaeocyates it's a very strange group because it started in the lower Cambrian, then it went to the middle Cambrian, and then disappeared. So it's a group of organisms that lasted for 40 million years and no trace of descendant. And archaeocyates are very interesting because they have some criteria which make them very close to sponges, and at the same time criteria which make them close to corals. So they are somehow they look like sponges because they have these let's say shape with pores. So they are like really poriferans, like sponges, but at the same time they have these radial walls here that you can see here, which make them very close to corals. So still nobody knows exactly. Seems that it has been a sort of aborted experience, biological experience with no descendant. If we go a little bit in the middle, at the end of the lower Cambrian, the beginning of the middle Cambrian, there is this Changjiang fauna, which really marks the beginning of the Cambrian explosion. And where you have about 150 species, 20 different phylum, and probably it's one of the first modern ecosystems. And this ecosystem has been well studied by a scientist in Lyon, Jean Vallier, who is really the expert of this Changjiang fauna. Let's go a bit upper, in the middle Cambrian. Well, no, it's already still the Changjiang fauna. So you see a lot of arthropods and some other groups here, poriferans, worms, priapulid, maybe some chordates, the first chordates, and so on. If we go to a bit later stage, in the middle Cambrian, we see the Burgess shell fauna, which is even more diversified, which has been, let's say, discovered at the beginning of the 20th century. And it's here you have like 37 phylum, 70% are metazoans with soft-bodied, but there are also some metazoans with skeleton, exoskeletons. And you find all kinds of phyla, from sponges to chordates and echinoderms. So here again, let's say some representative of the Burgess shell fauna. So in brief, if we sort of summarize what can have been the Cambrian explosion, it's probably the appearance of new body plants in metazoans. There was a net increase of diversity at all levels, from specific, generic, family levels, and so on. It was also the setting up of modern marine ecosystems. And you have also some aborted experience, and the archaeocyates, for example, here are really this type of aborted experiences. So what time is it now? Shall I stop for a while, or do you? 4:30. Shall I stop for 5 minutes? Because I'm really. I still have 2 hours to struggle. So
10 minutes.
2 hours speed and then another 2 hours.
Ah, okay, no problem.
Yeah, that's it.
Coffee.
Okay, I will take care.
Thank you.
I'll just continue this one, huh?
Yeah, yeah.
Because it's and half of the second. Also, I think super.
Facciamo prima finire le lezioni. Facciamo finire le lezioni.
So, everybody's here? Can I continue? Okay, so after this little pause, little stop, I'm going to just enumerate the different causes of the so-called Cambrian explosion. And this cause can be split or include some of more external causes, like tectonics, climate-related, cause related to oxygen, the seawater chemistry, and some are more internal, like physiological or predation of genetic causes. So I'm just going to enumerate these different causes. About tectonics, we know that in the late Precambrian, there was this supercontinent called Rodinia. On this continent, the supercontinent started to fragmentate, to be split into different subcontinents. And these, of course, induced the formation of new epicontinental areas. So people say that it may be, of course, cannot be a single cause for the explosion, Cambrian explosion, but at least it created some environmental conditions which could favor the Cambrian explosion. There are climatic causes, like the end of the snowball Earth, so the end of the glaciation. The end of the glaciation had different effects. First, it induced the increase of fluxes of nutrients into the oceans. It also increased the sea level, and it also promoted the formation of new ecological niches. And of course, the end of the snowball, we don't know if it's due to a massive release of CO2, of volcanic origins, for example, but there have been different scenarios hypothesized. What is interesting to notice is that there is a sort of correlation between this delta C13 curve, which is fluctuating during the Proterozoic, especially in the Ediacarian. So you have this fluctuation of this curve and maybe the appearance or the diversification of some groups. So it's difficult to go further, but at least there might be a sort of correlation between these events. But it's difficult to go further with this type of hypothesis. They are all the causes which are related to the level of oxygen. And the first paper, which is a very interesting paper from Dow in 1970, said that raising the level of oxygen could contribute to the formation of molecules like collagen. And collagen is interesting because some amino acids of collagens, like lysine, proline, are hydroxylated. So you get hydroxyproline, hydroxylysine. And for hydroxylating these amino acids, according to Tau, you need to get a certain level of oxygen in the, at least in the oceans. So we can even hypothesize that the Ediacarian fauna, small shelly fossils, then Cambrian fauna corresponded to different levels of oxygenation of the seawater. And here, this is what this graph more or less tend to show, that there is this progressive raise of oxygen in relation to the formation of, or to the diversification of animals. Some other causes are more related to the chemistry of the oceans. For example, there are studies showing that, for example, in Spitsberg, there were some hypercalcified bacteria in the Neoproterozoic, so just before the limit between the Precambrian and the Cambrian, and which induce or which suggest that the Neoproterozoic oceans were heavily supersaturated with respect to calcium carbonate. And some other hypotheses show that there may have been a decrease of the magnesium and calcium ratio. So this is the hypothesis of riding. And of course, you have also these ecological causes, like the appearance of predation. And I was mentioning traces of predation on the cludinid samples, but if you look especially in the Cambrian deposits, there were lots of traces of predation on the trilobites. So predation may have been a sort of trigger for animals to form a hard part. And we know, of course, that in the Middle Cambrian, there were some killers like Anomalocaris, which was the super predator of its time in the Cambrian seas. But I'm not sure that this is enough to explain the appearance of calcium carbonate skeletons. Of course, you have also the genetic causes. We know that there was this diversification of life, especially the diversification of the phyla, and which may have been related to also the formation and diversification of developmental genes, like the Hops genes. So as you may know, the Hops genes they code for transcription factors, and they control the expression of other genes. And if you look at this sort of this graph, this phylogeny, let's say, based on the Hops genes, you see that there is a diversification of the Hops genes from sponges to vertebrates, and which suggests that all these genes have been evolved by duplication. So with a quick evolution of these genes, let's say, promoted the evolution of life. Well, there was also an hypothesis that we sort of invented about 30 years ago together with Peter Westbrook, and we called it the anti-calcification scenario, where we were in if you imagine the oceans of the Neoproterozoic, so at the end of the Proterozoic, so before the Cambrian explosion, oceans were supersaturated, and metazoans needed inhibitors to avoid overgrowth of their tissue by crystals. So they may have used substances like mucus substances that, let's say, secreted on their tissue. And these mucus substances may have been reused at the limit between Precambrian to Cambrian, not for inhibiting, but for acting as the opposite way, meaning controlling the calcification. And that was a sort of crazy idea that we had, and which we published about 30 years ago. So what is sure is that to go from a system where you have only naked animals, so animals without any shells, any exoskeletons, to animals of modern ecosystems, which can be partly or completely mineralized, you need really a different, let's say, factors that I explain, especially when I spoke about the biologic control mineralization. And you need first to make a hierarchy in the structure, and you need also specialized organic matrix. And this part was, of course, very important. That's probably one of the most important events in the history of life on the Earth. So the accession of the biologically controlled mineralization. And then it was followed during the Paleozoic time and the Mesozoic and Cenozoic by different events, which were also sort of important in themselves. During the Paleozoic times, if we sort of summarize what happened, we said between the Cambrian and the Permian, so that's between 542 million years ago and 250 million years ago, the calcium carbonate was dominated by very heavy reef systems, mostly in a calcitic ocean. And you have these what we call these Paleozoic corals, like tabulate corals, rugosans. You have also stromatoporoids, and all these corals, which were which could which developed intensively, especially in the Ordovician, in the Devonian, in the Carboniferous, and also Permian. They got extinct at the Permo-Triassic crisis about 250 million years ago. So what happened later with the secondary era, what we call the Mesozoic? You had the emergence of planktonic organisms, which were important providers of biogenic calcium carbonate. And we can speak again about the case of coccolithophore algae. So coccolithophore algae, they were really, they started to be present in the Triassic period, but of course met a huge development during the Cretaceous period. And of course, we all know these examples where you have these chalk cliffs made of just coccoliths, like in Normandy, like in England. And to have these huge deposits of chalk, we must have had a sea which was completely full of these organisms. And it's very difficult to conceive nowadays, actually. But there must have been a burst of these animals in the seawater in the Cretaceous times. Again, another accumulation of coccoliths in the Sussex, in Seven Sisters. There you see these huge amounts of coccolithophore algae in these cliffs. And today, here is the map taken from a satellite map of Alaska, where you see here a huge bloom of coccolithophore algae. So you have Alaska here, Aleutian Islands, and here you have this huge bloom, which is of more or less the size of Alaska. So it means that even today you have these huge blooms. Of course, they do not have the amplitude of the blooms in Cretaceous periods, but still they are very important provider of calcium carbonate. And during the Mesozoic times, we also had this fluctuation between what we call calcitic seas and aragonitic seas. And this is what these curves show, especially in red, where you have calcitic seas. And the ratio between calcite and aragonite is fully determined by the ratio of magnesium. So if you have a lot of magnesium in the ocean, if you have a high ratio of magnesium on calcium, you will have an aragonitic sea. If this ratio is low, you will have a calcitic sea. And you have this huge fluctuation during all the Mesozoic times, especially during the Paleozoic times. Most of the Paleozoic was calcitic seas, but then in the Carboniferous here, Devonian, Mississippian, in Carboniferous, you had these fluctuations started, and then again the calcitic sea in the Cretaceous period, which may also explain why you had these such blooms of coccolithophore algae. Some other things that happened during the Mesozoic times is that while calcifying planktonic organisms emerged, especially coccolithophore algae, but also diatoms for the silica world, you also had the emergence of new reef systems made of scleractinians that continued to develop during the Triassic, the Jurassic, and the lower Cretaceous. And then there was this, in relation to what I showed before, is this calcitic sea. You had a group of organisms which took the lead, which took over, especially in the upper Cretaceous. And these organisms are called rudists. So they are really bivalves, big bivalves, making these huge reefs for kilometers, and which can be one of these rudists can be like more than 1 meter high, and they are massively producing calcium carbonate. And you have these reefs making big bars of big layers of calcium carbonate. So, in short, the message the takeaway message would be that well, you have the biologically induced calcification, which started with the stromatolites, which was very dominant during the Archean and also in the Proterozoic, before regressing, but not disappearing, just regressing. And then the appearance of biologically controlled mineralization that heavily started in the Cambrian explosion, 444, 544, 42, 43 million years ago, which particularly was effective for benthic fauna and neritic domains. And then something that happened later, what people call the Mesozoic revolution, or during especially the Jurassic, there was a sort of takeover of the calcification by planktonic protists like coccolithophore algae, and this happened more in open oceanic domains. So now I go to the maybe the third. No, no, the fourth.
Ah, fourth.
Sorry.
Yes.
Yes. Thank you. So I'm going to sort of more specifically speak about the mollusk shell formation and the mollusk shell biomineralization. Let me introduce the mollusk as a phylum, as a group. You know, in the metazoan world, the mollusks represent the second biggest group, far after the arthropods. The arthropods is between 1 or 2 million living species, maybe more. The mollusk is only 120,000 living species, but still, it's twice as much as vertebrates, probably even much more. And the mollusks are actually divided in different groups, in different classes. I will just mention these very anecdotic classes called solenogasters, codophagoates, which are mollusks which do not have a shell. They have calcified teguments made of little needles of calcite, let's say calcium carbonate. You have also the polyplacophore. You probably know that under the name chiton, the chiton. Polyplacophores, they have these sort of shell made of plates, It's a rather small group, about 900 species. And you have also a group, very peculiar, called the monoplacophorans, so which are represented by only 15 species, and which was discovered as a sort of living fossil in the 50s, I would say. And the monoplacophorans are really supposed to be the ancestor of all the mollusks that have a shell. But of course, we will more concentrate on these groups. First, the bivalves, with about 12,000 species. The scaphopods, they are not very known, 400 species. The dentalium, those tusk shells. So many people do not know them, but the gastropods, with more than 100,000 species, which is the biggest group in the mollusks, and also the cephalopods with the nautilus. But of course, keep in mind that in the past, cephalopods were very dominant, especially during the Mesozoic time with ammonites. Ammonites were extremely abundant and a lot of species. And now the cephalopods are a bit more, especially the cephalopods which have a shell. It's mostly nautilus. You have cephalopods with internal shells, like cuttlefishes. And also you have cephalopods which have completely lost their shells. So these are the octopus. So again, the mollusk, they as a group, they appear very early. They were really the witness of the Cambrian explosion, especially the monoplacophorans, the bivalves. So they very early in the lower Cambrian, there were traces of these animals, especially in the small shelly fossil SSF fauna. And to speak very generally, since the Cambrian, the mollusk have a complete continuous fossil record. And this group has a great adaptability. They have also a very high evolutionary success. And this success is very easily due to the shell in itself, because it's an efficient shelter against predation, desiccation, and it can really contribute to the colonization of several environments. Keep in mind, for example, gastropods, they have colonized all the environment, so caves, high altitude, also very cold environments, hot desert, so they are everywhere. So they are they are actually the most successful group in mollusks. So let's concentrate on the shell. So the shell is a multi-layered material. And you have here, when I say multi-layered, it's only few layers. There are not many layers, but it's a layered material. And I show here the example of a freshwater bivalve. It's called a freshwater mussel, that's very common everywhere in Europe. And if you cut this shell and you look in the section of this shell with a scanning electron microscope, you will see different layers. So the outer layer, the one which makes the shell a bit greenish, like olive colored shell, is the organic layer I was speaking about a couple of hours ago. It's the perostracum. Below the perostracum, you find here an external layer which is calcified. In that case, it's made of aragonite, long crystals called prismatic aragonite. And internally, you have another layer made of very tiny crystals that we hardly see here, and this is the nacreous layer. And all these two layers are calcium carbonate. Here it's fully aragonitic, but keep in mind that some animals can have an outer calcitic layer, inner aragonitic layer. Some other animals can have a fully calcitic layers, like edible oysters. So again, about the physiology of calcification, I just remind you what I've shown before, that what makes the shell is the mantle tissue, the calcifying mantle. And the mantle has a very complex chemistry because it allows the concentration of calcium ions or bicarbonate. It allows to extrude these ions, but in a sort of ordered way, together with the secretion of the matrix. So the ions can be extruded probably in ionic form or under amorphous granules. And of course, if the system has to work, somehow you need also to reabsorb protons. You probably have some proton ATPases or proton pumps that remove the H+, the protons, to allow the system to go to the production of calcium carbonate. Again, I will not comment this in detail. You have the mantle here, the perostracum, which isolate the compartment from the outer part of the environment, and the growing shell here. And the shell is actually what we call the organomineral material. Why? So if you take a shell, you dissolve it into a solution of acetic acid or vinegar, say, you will be able to dissolve entirely the shell, but you will also release some organic components. And these organic components, they are what we call the organic shell matrix, and they are constitute 0.05 to 1 or 2% of the shell. So most of the shell, say 99%, is calcium carbonate, and you have grosso modo 1% of organics. And these organics, they are proteins, peptides, polysaccharides, lipids, pigments, metabolites, and there is sort of gradient of from very well studied chemical species like protein to a very poorly studied. So actually what we mostly know of these shell matrix are the proteins associated to the shell. And it has been now already quite a long time that mollusk shell matrix is really considered as a toolbox, is a molecular toolbox for the deposition of mineral by mollusks. And it's supposed to display major roles in biomineralization. But of course, since the promotion of the idea was that the matrix was associated with the mineral deposition, so since that period, there have been a very drastic evolution of the concept of skeletal matrix in the last decade. And I will show you here a little bit of the evolution of these ideas. So in particular, the manner we perceive the shell matrix and its putative function has a lot to do with the technique we use to analyze this matrix. And I show that now. In this graph, I represented the different approaches that scientists used to study shell matrices. First, we made the story start from the after-war period, so the 50s, where you had the development of the biochemistry as a main discipline. And as I said, biochemistry was very good for characterizing shells, for characterizing shell proteins, but in a very specific way, meaning you cleaved all the shell proteins in amino acids, and then you could make amino acid analysis. But then came molecular biology. So especially in the 80s, which started to develop and then was prominent in the 90s, for example. Mm-hmm. And after that, the third approach was the omics. So, I mean, the high throughput screening techniques that allow to, let's say, get in one time, all the components, all the protein components of the gel. So let's go with the biochemical approach. As I said, most of this approach was based on amino acid analysis, but also you had chromatographies or gel electrophoresis. And when you were lucky, you could even get some partial N-terminal sequences of proteins, maybe 5 or 10 amino acids, and that was, you were happy. You had reached this stage. And of course, this was very limiting somehow. This approach prevailed a lot in the 60s, 70s, 80s. And but then molecular biology came. So with molecular biology, instead of looking directly at the proteins, you look at the gene that encodes the protein. And by using molecular biology technique, it was then possible to obtain full sequences of proteins, to do some in silico work, so to work with the sequences and to try to get all the information by using the sequence. But I would say that this approach was very, of course, prevalent and pertinent, but it was a one-per-one approach. So meaning that you got one gene encoding one shell proteins, and you try to make everything with that gene and that protein. Try to find some function of that protein and so on. But things are a little bit more complex. We deal with complexity, of course, and going one per one is a little bit limiting also. So that's why these omics, these high throughput screening techniques were set up, especially in the year 2000, 2010, where by using genomics, transcriptomics, and proteomics, and especially combining transcriptomics and proteomics, we could get the full protein content of a shell. And that was a very big improvement. To go back to the biochemical approach, this is the period where people said that the protein were behaving as a mold around crystals, or maybe a sort of physical template. Some hydrophobic proteins could act as a sort of mold, but there were also the discovery of some proteins which were polyanionic, so very negatively charged. So we have to think that negatively charged proteins means that these proteins are able to bind calcium ions. So there were some the development of some models, like I show here. And these models were emerged in the mid-80s, where you have, for example, this ternary model, where you have chitin fibers. So remember that chitin is the polymers of N-acetyl glucosamine, so that's a very long sugar, making these sort of layers, which is a sort of structural support. In this model, you have also some very acidic proteins which are soluble and which allow nucleation and inhibition. And you have some insoluble proteins which act more as a mold or template or even an hydrogel. So about 24 years ago emerged this very interesting model for explaining the, especially the Naker model of pearl calcification. With these three ternary systems, so chitin, acidic proteins in red, and a gel made by these insoluble hydrophobic proteins. So that was 25 years ago. Then with molecular biology, as I said, we had full sequences. So people started to publish sequences of different proteins. It went one per one, so like going very, very slowly. And I show here a few examples of proteins that were discovered at that time. For example, the first published protein, shell protein, mollusk shell protein found or identified was nacrein. It was the paper of Miyamoto in 1996, where you have this protein made of different domains. And some of these domains are enzymatic domains, or they are supposed to be enzymatic domains. So this protein was a little bit different from what the model predicted. We started to see that things were a little bit more complex. Then you had the lustrin A, which was published in 1997, which is a very complex proteins, very long proteins with an alternance of cysteine and proline-rich modules, what you call a GS loop, so glycine serine loop, and so on. In 2000, I published the mucoperline, which is another type of proteins. It's a mucin type of proteins with tandem repeats and domain. So all these proteins do not really fit into the models which were proposed at that time. And of course, going a bit further by using these large high throughput screening techniques, which allowed the obtaining complete shell repertoires, we saw that things are even more complicated because we identified a lot of molecular functions. I just show here how the omics approach works. Suppose you have a shell, you have an animal. Here it's Magallana gigas, so it's the edible oyster. So instead of eating the oyster, we use it for making science. And on one side, if this animal is supposed to calcify, we have to let it calcify, of course, in a basin, and you can sample it very quickly and detach the living calcifying tissue here. From this tissue, you can extract the messenger RNA that you can convert to cDNA, and you can massively sequence these cDNA, and you obtain finally a transcriptome of the mantle tissue. So the transcriptome is actually a sort of instantaneous picture of all the transcripts, all the messenger RNA that are produced at a given level. So it means that you really need to have your animal actively calcifying its shell for getting all the good transcripts. And at the same time, you can take the shell, and of course, you have to clean it and so on. You can extract the skeletal matrix by dissolving the shell, and you make an enzymatic digestion and make this mass spectrometry, this tandem mass spectrometry analysis. And by combining the transcriptome and the data of the MSMS analysis, you can really get all the protein, all the transcripts that are encoding proteins that are found in the shell. Okay, and after that, there is this. You get this what we call the skeletome, so the occluded proteins, so the skeletal repertoires. And nowadays, they are. I think this table is probably not valid anymore, but we know some data for more than 25 genera of mollusks, mostly in bivalves and gastropods. And so we have, of course, a very partial view of the composition of the protein composition of the shell, but still it's much better than what we knew 20 years ago. So the contribution of the omics technique is the following. So there is a massive increase of the number of identified proteins. We know that the concept of skeletal matrix is a bit blurred, and I will explain you why. We have identified several new functions. A majority of these functions are unknown, and they are novel, so it's difficult to go further. We don't know yet if all those proteins that we find in a shell, they are really contributing to the formation of the shell, or if they are contaminants or doing some other functions. What I can show here is that the number of shells, if you remember, I was speaking about nacrein. Nacrein was the first protein discovered in 96 and published in September 96. And here we have much more than thousands of proteins identified to date. So it's really, we have a burst of the data concerning mollusk shell proteins. And I sort of summarize here what we feel, or at least what I feel, about shell proteins. So here you have the calcifying tissue, the calcifying epithelium of the mantle, which is supposed to secrete a lot of proteins. Some proteins may not be involved in biomineralization. Some of them may be involved. They may be occluded. Some of them may be not occluded. But we have also, and that's what we discovered by doing proteomics, that we probably have a cleaved extracellular domains of membrane-bound proteins. Some of these cleaved domains may be occluded and some not occluded. And you may have also some cellular contaminants, which might be occluded and not occluded. So it means that we have a very complex picture now of the skeletal matrix. And again, we don't know if all the ingredients of the skeletal matrix are associated to the mineral deposition. So to summarize, the facts are we know much more than 1,000 proteins. We have several functional domains. These domains are belonging to several families. And we focus our attention on what we call the low complexity domains, because this is a very common theme that we find everywhere. So these are the domains that are dominated by 1 or 2 or 3 amino acids. You may have some domains that are, for example, polyaspartic domains, or domains enriching glycine and serine residues, so they will make GSG, SG, SG, and so on. So all these domains are very peculiar, and they are very abundant in shell proteins. So the problems that we have now is that the concept of skeletal matrix is a bit blurred. We still have problems to relate protein sequence to functions. And of course, we don't know how all these proteins work together. So the possibilities that we have now is first to compare between different calcifying repertoires, to try to sketch some macroevolutionary trends, and also to work on, I will come back at the end of my the day, last 30 minutes, for the diagenesis of skeletal matrix proteins. But I want now to focus on a few examples, a few scientific questions that we had. And one is related to the invention of nature. Nature, also called mother of pearl, is something which is very specific to mollusks. We don't know if it was a single invention or it was invented different times. I show here different animals, so some bivalves like the edible mussel, the freshwater mussel, the pearl oyster, and another animal, another bivalves here. And they are representative of different groups, and they all have an internal nacre layer. You have also here a gastropod, the abalone, Haliotis, and here you have Nautilus, the Nautilus, and they also are nacreous. So if you look at more specifically the nacre in mollusks, you have, for example, the brick wall nacre that is very common in bivalves, which can have different structure, but still the theme is common to all the biomes, where you have these flat tablets, and they are made like a brick wall, which is sort of symbolized here by this drawing. So these flat tablets are these black parts here, and they are really disposed like a brick wall. So, which means that if you want to propagate a crack here, it will be difficult because of this structure. So this is for bivalves. Now we have this a little bit something slightly different for gastropod. This is what we call the columnar nature, where the tablets are a bit more aligned, but of course there is always a shift between one layer of tablets and the other, so that you still maintain this difficulty to propagate a crack. But you see that the tablets are a little bit more aligned vertically. Huh? So this is the columnar nature in gastropods. If you look now the distribution of nacre in mollusks, you see that nacre is or was present in monoplacophorans. The scaphopods, they do not have any nacre. Bivalves, nacre is present in different orders, like the Anobanodexmata, Paleotherodon, Pteriomorph, Paleotaxodon, bivalves. So the nacre is present in at least 4 different orders. In cephalopods, nacre is present in nautiloids. And in gastropods, nacre is especially present in vetigastropods, which is one of the big group of gastropods, like the abalone. The abalone is one of the representatives of this group. And within nacreous bivalves, we made some comparison between 3 different orders: unionoids, pterioids, and metyloid bivalves. And we saw all these, when you look at all these names are names of proteins. And we saw that these 3 types of nacre have some similarities, but also some differences. It's a very, you see here, for example, for unionoids, you have a complete set of proteins which are unique to the unionoids that you cannot find in the pterioids or metyloids. So you have both shared proteins, but also proteins which are really unique to each of these groups. Okay. Now there was this comparison that one of my former colleagues, Benjamin Marin, made when he was doing his postdoc. He made between the pearl oyster nacre and the abalone nacre. And if you try to compare the proteins associated to these 2 types of nacre, there was only 1 protein in common. And the others were the other proteins were absolutely different. Which so in one case, the pearl oyster has a sheet nacre, while the abalone has more a columnar nacre. And it seems that in that case, it means that, or it suggests, of course, strongly that these 2 nacres are just built from different tools, from different molecular tools, from different protein tools. So it's very interesting because the first conclusion that we had, maybe it has to be modulated a little bit more, but still. So within bivalves, we saw several similarities, but also taxon-specific nacre proteins. And between bivalve and gastropods, we found some very different nacre protein assemblages. So this may suggest, and I'm very careful about that nacre may have been invented at least 2 times or maybe 3 times. So it's not a unique invention. But it might be also that a part of the proteins that are associated to the deposition of nacre have a rather fast evolution. So maybe from common theme, ancestral nacre, these proteins evolve in a completely independent manner. So they with a drift from a common nacre ancestor. But what is interesting behind this, is that it seems that different assemblage of proteins can build similar structure. So it means that the system has a relative plasticity. Yeah? If you take different proteins, different protein assemblage, and finally, they will drive to the same type of microstructure like nature. Of course, if this is true, now the next question is, which is the driving force that constrains the system to produce nature? And this, we don't have the answer. Another question that we had was the relationship between the, let's say, protein assemblage of prism and nature. And there is this nice example of the pearl oyster. It's not an edible animal, sorry, but the pearl oyster, Pinctada margaritifera, the pearl oyster from French Polynesia, which has this very nice, it's very nice shell, very heavy shell, very solid. The internal side of the shell is made of nacre, and the external side is made of prism. In the case of nacre, this is aragonite, like every nacre. All the nacres are made of aragonite, while the outer layer is made of calcitic prism. So if you make a cross-section of the shell, you will see the nacre here, which is also symbolized here. So it's with the brick wall nacre, this very flat tablet. The thickness of each tablet is 0.5 micron, so half a micron, and it can be about 10 microns in length. And here the outer layer, the prismatic layer, is made of very long elongated crystals. So completely different microstructures. So the idea was to are the prism and nacre made from the same protein assemblage? Actually, that maybe not. Keep in mind that this sort of very simple question was kind of fundamental, especially in the field of biomineralization, and it was left open. This question was left open for decades. And I show here a few examples of papers, like the paper of Philippe Hare published in Science in 1963, where Philippe Hare showed that the amino acid, then the amino acid analysis, amino acids of the proteins from aragonite and calcite were a little bit different. The proportion of amino acids were, and that was in the model of Mytilus californianus, which is an edible mussel from America. More recently, but still an old paper from '67, Charles Grégoire published this paper on the structure des matrices organiques des coquilles de mollusques, where he also showed that there might be some differences, and also Steve Weiner in biochemistry in '93. So it means that this question about prism and nacre proteins, the matrix associated to prism and nacre, this question was sort of open for since at least the 60s. So we did this combination of transcriptomics and proteomics, and we found these assemblages of prism proteins here, and here nacre proteins, and the result was that the protein composition were very, very different. And this was published 13 years ago in PNAS, where we looked at the different nacre and prism proteins and looked at their supposedly functional domains, and we could, let's say, enumerate the functional domains. And if you look at the intersection between the prism repertoire and the nacre repertoire, we had very, very few proteins in common that were shared by the two layers. So meaning that for making nacre, it's quite different than for making prism. So it means that, in other terms, the calcifying mantle has its own functioning for making the prism protein, for making the prismatic layer. And the same mantle, so if you go a little bit more inside, the naked, the calcifying mantle has another functioning for making nacre, for depositing nacre. The third example is about the mosaic composition of shell matrix protein. And this is a work from our colleague Daniel Jackson and his group in Göttingen, and we also contributed to this by doing the proteomics. And it's about the freshwater pulmonate gastropod, Limnea stagnalis. Limnea stagnalis, it's not at all nacreous. It has a completely different shell microstructure. It's called cross-lamellar structure. And we identified at least 40 proteins, which were rather abundant. And Daniel Jackson's group, for each of them, they could really make in situ hybridization and see that each protein was associated to a certain area in the mantle tissue. What is interesting is that, so in this circular diagram, suppose that all these little stripes that you see here, each stripe is one protein of the shell of Limnea stagnalis. And when you have these long, let's say, lines are the correspondence between the proteins of Limnea stagnalis and proteins of other models. So here you have echinoderms, cnidarian, some gastropods, and so on. So you see that most of the proteins of Limnea stagnalis are rather close to the proteins of the gastropods Sepae nemoralis, which is the land snail. You know, that makes sense, actually. But you also have some similarities with proteins of Pintada margaritifera, with Mytilus coruscus, Crassostrea gigas. So it's a very patchy composition, actually. So you have a similarity of the proteins of Limnea stagnalis to many different proteins of other invertebrates. So we have this sort of mosaic picture. So to sort of summarize about especially the shell proteins, we can say that if you take one shell, in one shell you will find from few dozens to more than 100 proteins in a shell, different proteins. Many of them are modular proteins. Some of them are even some chimeric proteins. So each of them is supposed to be kind of multifunctional. And the function of these proteins are mainly deduced from sequence analysis, which is, of course, a limitation. And there are very, very functional assays, like gene knockdown. So very, very few things have been done on these proteins. If we want to sort of segregate or split the proteins into two domains, or two not domains, but two sub assembles, subset, we see that some domains of these proteins are clearly identified as enzymatic or sugar binding, protease inhibitors, calcium binding, immunity-related or extracellular matrix-like domains. But you have a huge bunch of domains that have unknown or only putative functions. And among these domains, we have these domains of low complexity or repetitive low complexity domains. And these are the domains for which we have very poor ideas about their functions. So I think this is, to summarize, the skeletal matrix proteins in mollusks, huge diversity, certain plasticity of the system, modular architecture and sequences, abundance of low complexity domains or repetitive low complexity domains. The macroevolutionary trends for skeletal matrix proteins is we can say a few things. In particular, there is a complex evolutionary histories. Some of these proteins may have been recruited very early, like in the Cambrian, for example, so more than 500 million years ago. But these proteins evolved, and you have a lot of secondary evolution in the different mollusk lineages. And some of these proteins may have been recruited very recently, and they are more lineage dependent. And some of these proteins may have also, as well, fast evolution. So if you take the main message, is that there is no real phylogenetic signal given by these shell proteins. So we have to be very careful by using these shell proteins for doing some taxonomic work, for example. I have a last. Okay, so I will finish at half. So we can, yes, diagenesis.
Okay.
Okay, are you still alive? Okay, not you are not completely fed up. Well, you may be fed up, but you don't say it, of course. Okay, so this is my last talk. I should last until 6:30. So now we're going to speak about something a little bit different from what I've shown before. This is what I call the shell matrix diagenesis. So who knows a little bit about what is diagenesis? Have you heard about this terminology? Are there people who have done earth science or sedimentology or so diagenesis? Does it sound familiar? Okay. Yes, more or less it's like that. Yeah, it's diagenesis is really the transformation of sediment into a rock. And you can apply the terminology of diagenesis also to, of course, living system like shells. Shells, they will transform. So when the animal dies, the shell will bury into the sediment, and the shell will have its own evolution. So this is what we call shell matrix diagenesis. Again, I start with very simple ideas. Whatever calcium carbonate you, let's say, skeleton you use, like cnidarian, bryozoan, brachiopod, mollusk, echinoderms, if you decalcify these calcium carbonate biominerals, you will be able to extract a little bit of organics. Huh? I told you that organics are less than 1/10 of a percent till 1 or 2% of the skeletal weight. So it's not a lot. If you look now, if you, in my presentation, there are always some reminders of the other presentation. So if you look now, something that I've shown 1 hour ago, 1 or 2 hours ago, in the neritic environment, you have this benthic production of biogenic calcium carbonate. I said that it's about 2.5 billion tons a year, okay? One third of which is made of from corals. If you look in terms of skeletal matrix, let's say skeletal matrix is also modo 1% of each skeletal tissue. It means that every year you produce, it's visible, between 1.2 million tons a year until 50 million tons a year of skeletal matrix, which have the possibility to be buried and to be to reach the fossil record. So that's quite a lot, actually. So 1% of this huge production of calcium carbonate, 1% of this can potentially be buried and joining the fossil record. So the question is, how do these molecules, which are occluded in shells or in skeletal tissue, how do these molecules fossilize? Again, we have to make a bit of history. And I cite here few papers, especially the one of Abelson from 1954. And it was published in a Carnegie Institution of Washington yearbook. And the paper is called Amino Acids in Fossils. And that was, to my knowledge, one of the first papers mentioning that in fossils, in calcified fossils, you had amino acids. And Abelson went further and he published another paper, among which one called Some Aspects of Paleobiochemistry. And paleobiochemistry was actually a discipline that emerged from this work of Abelson. And at that time, there were two approaches to tackle the problem of fossil proteins and fossil organic remains in shells or in skeletal tissue. Either to work directly on the fossils, so that meant extracting organic material and analyzing organic material. And at that time, remember what I said at the beginning of my talk, that was mostly amino acid analysis. So you had to extract and cleave everything in amino acids, complete small pieces, and then do some chromatography, and then say, okay, in this fossil, I found 10% of glycine, 15% of aspartic acid, 20% of alanine, and so on. That was how it worked. And there were also the possibility to do artificial diagenetic experiments. So you take a shell of today, living shell, you kill the animal and then you take the shell and then you heat this sample, you heat the shell, you extract the matrix, and you see what remains from the heating experiment. To go again back to history, I would say that the golden age of fossil protein analysis was really started in the 60s until the 90s. And there was this very interesting book published in 1980, Biogeochemistry of Amino Acids, where it's a multi-author book where there are different models, it's different systems like shells, but also bones, and where lots of analyses are made on fossil material. And of course, I speak about the golden age of fossil protein analysis, because this golden age came to an end because of what? Because of discovery of PCR, polymerase chain reaction. And with PCR, you can amplify DNA. And DNA, which does not have a high potential for preservation, still can be analyzed from subfossil or fossil material. And the use of PCR, of course, opened the door to the analysis of fossil DNA. And as soon as the fossil DNA started to be analyzed, people tend to forget about fossil proteins, because you cannot, that's one of the weaknesses of the approach, you cannot amplify fossil proteins as you can amplify fossil DNA. So somehow the age of the golden age of fossil protein analysis sort of went down, but there are some good perspectives for the very close future. So what can happen to a fossil protein? First, it can be hydrolyzed, partial hydrolysis. So you go from proteins to peptides and peptides to free amino acids. And when, of course, you cleave your proteins entirely in amino acids, then you lose entirely the message which is carried by the protein. What can happen also is that you have racemization of amino acids. So you know that amino acids are chiral molecules, so they have this chirality, and somehow by racemization they go from the levogir conformation to dextrogir conformation. That's also physical, let's say, physical transformation at the amino acid level. You can have also what we call defunctionalization. So you go from amino acids. Amino acids are acids with acidic function and amine function. So after a while, one given amino acid tends to lose its amine function and sometimes tends to lose its carboxylic acid function. So you decarboxylate or you desaminate your amino acids. So you end up with amines, organic acids, or hydrocarbons. And then when you are at that stage, there is no more signal that you can exploit for trying to see what was the initial proteins. And finally, what can also happen is that you have condensation. And these reactions are actually the reactions of amino acids with sugars. And they can make this very complex material, these unique acid substances material, very complex geopolymers. And when they do like that, then you lose the signal as well. About the hydrolysis of skeletal matrix proteins, it's really a multi-parameter story because it depends on the peptidic bonds. And to give you an example, the peptidic bond between one glycine and one alanine is about 100 times stronger than the peptidic bond between two aspartic acids. So it means even peptidic bonds have different stabilities. Okay. It depends also on the structure of the protein, the 3D structure. If the protein is a sort of coiled or not coiled, is it globular or not globular? It depends also a lot of the post-translational modifications, glycosylation, phosphorylation. It also depends on the localization of the proteins in the biominerals. So here in this very sort of simplified graph, I show that some proteins are intercrystalline, so they are in between crystals, but some other proteins are inside the crystals. And the proteins that are inside the crystals, if the crystal do not recrystallize, it means that we have a good chance that these proteins will be preserved. But it won't be the case for intercrystalline proteins, like here. For example, here, these proteins can be very easily degraded. So the localization of the proteins in the biomineral is very important. What is also important is the presence of water. So if you have interstitial water, if the water is linked to the mineral or not, so if you have interstitial water, water can just help degrading the proteins. And also it depends on the polymorph that you consider. For example, calcite is a very stable polymorph of calcium carbonate. So it means that calcite, if you bury calcite in the sediment, you have a good chance that after 50 million years, your calcite will remain like that. But for aragonite, it's a different story because aragonite is metastable, so it has a chance to retransform, recrystallize into calcite. So meaning that the mineral that is considered can have its importance for the preservation of the shell or of the calcium carbonate skeleton. So we've done some different examples, and one of the examples I would like to show here is the thermal stability of naked proteins. And we did some artificial diagenetic experiment where we take the naked powder, we homogenize the nacre, and we put nacre into little tubes, and we heat the tube at 100 degrees for 10 days, well, for different days. And every day we remove one tube from the oven. So we sample every day, and after that we extract the matrix, and we try to quantify the matrix by different systems, different techniques. For example, by using antibodies, and I don't know if do you know what antibodies are? Yeah, so they are proteins of the immune system. They are proteins which can retarget specific proteins. So suppose that you have antibodies made against naked proteins. Well, if you suppose that your protein is here, and it has been extracted from a heated sample, and it's a little bit transformed, your antibody will not retarget very good your protein. So if the protein is not transformed, if the protein is intact, not transformed by diagenesis, then your antibody will strongly bind your protein. So here, by using different antibodies, and we are at T0, so no heating until T10, so 10 days of heating, we saw that there was a sort of decay, but very slow decay of the reactivity of our antibodies against the matrix proteins, the naked proteins. So it means with this system that we had already a first feeling about the slow degradation of the naked proteins. Some proteins may be degraded, some of them not. But so we had this sort of overview of what may happen in our system. For example, after 10 days at 100 degrees, we lost like 20% of the reactivity of the antibodies. Then we did proteomics. Proteomics, I said, and also you take your sample, you analyze it on the tandem mass spectrometry, and you can really see the proteins that disappear. So for example, at T0, we had different set of proteins here, which were the numbers in red are the numbers of peptides identified in the proteins, okay? So for nacrein A1, we have like 13 peptides. So after 5 days of heating, so that means 120 hours of heating at 100 degrees, we could see again nacrein A1, but there was only 7 peptides identified, okay? And after 240 hours of heating, so at 10 days, we still could find nacrein A1, but it was only recognized by 4 peptides. So it means that we could recognize our proteins after this long heating, but a part of the protein may have been degraded, okay? And the same if you look different, like PIF. PIF, it's another proteins, here recognized by 11 peptides. It's only 3 peptides and 3 peptides. And some other proteins, like purlin, we don't see it anymore. And here, USP11, 1 peptide, and then we don't see it, and so on. So it means that what we saw is that there was a persistence of some shell proteins, even after 10 days of heating. There was a sort of apparent degradation of some others. So there was this differential degradation. And the diagenesis was seen here by using proteomics by 2 characters, so the deep disappearance of some proteins and the decrease of the number of identified peptides. So another example, which is very closely related, we had some subfossil tridacna from French Polynesia, and again, so these were heavily, these were really well dated. I mean, the dating was very, very good. And the context was in one atoll, we had these specific diagenetic context. It's a flat, rich in percolating water, and it's not favorable for good preservation of aragonite because all these animals are made of aragonite. So we did some controls of the mineral phase. You could see that the mineral phase was attacked in many places, but still the microstructure looked fresh and not too transformed. Okay, and we did some analysis, proteomic analysis. I go directly to this sample. For example, here we have a sample of nowadays, so it's not a subfossil sample, for which we have two proteins that are defined by their peptidic signature. And here, if you see the subfossil proteins, you see that some of the peptides are not identified anymore here. The red peptide here is not identified here. So the coverage of the protein sequence is reduced in the subfossil protein. The same for this protein called Trinity and so on. You see that the coverage goes from 30% to 11%. And we have also a very big protein here for which we have a complete coverage of the protein of the living shell. And if you look at the fossil shell, you see that the coverage is very low. So it means that probably many of these peptides in red are degraded or at least not recognized by the mass spectrometry analysis, the proteomic analysis. So it means that here again, we have an idea that diagenesis affects, of course, some proteins, and some proteins can still be recognized by proteomics, but not entirely. So meaning that some domains may be destructured or cleaved or. And the third example, and third and last example, is the one of the last example is it's about archaeological sample that we treated some years ago. And it's a very, very beautiful example. It's a school case, I would say. And it's about what we call double bottom pearls, also called Doppelknöpfe in German. So these Doppelknöpfe are tiny buttons made by ancient tribes. They are very tiny, and this size, not more. You have to imagine that they are made from shells. So imagine that these people of 5,000 years ago, they had to cut shells slice, and then each slice had to be cut like that to make sort of little cubes. And these cubes were rounded, so probably by polishing. So imagine that they had to polish like that. Then they didn't have polishing machines. And then, with a little thread, they could make a little hinge in the thickness of the shell. So you have a shell which is about this thickness, few millimeters in thickness, and in the middle of the shell, they could have a little thread, a braiding thread, and so they could make this little groove. This little groove. And why did they do that? Because they have their leather suits. They were piercing holes in their suits, and then they were putting these little shells, these little buttons, which were just because of these of the groove, they could be inserted in the hole without falling. So that was very clever. So imagine that they could remake complete clothes made of these shells, and something like this here, which I show here. And it's funny because if you look in England, you have the tradition of the pearlies. I don't know if you've seen that already. So people who have their clothes completely covered with naked buttons. And this is what I show here. And actually, it's the pearl is sort of reminiscence of this very old tradition of the Doppelknöpfe of 5,000 years ago. So we had these archaeological samples, and 3 sites. They were identified in 3 sites. One in Denmark, the other one in South Germany, or South Germany, I think, or north of Switzerland. I don't remember. And the other one in Romania. And what is funny is that these 3 sites have exactly the same type of Doppelknöpfe. Okay, and they are more or less of the same age. Yeah? So the idea was actually there was this prevailing idea that these Doppelknöpfe were actually made from shells, which were transported from Mediterranean Sea to the north, to the east. So very long exchange, commercial exchange of shells of the late Neolithic, and that was the idea. So we did some experiment, especially Jorone, who did a PhD with us, and also Beatrice. So we had this archaeological sample. First, we did some analysis with scanning electron microscope. We saw that these Doppelknöpfe are really made of nacre. So this is typically a brick wall nacre. So this is bivalving nacre, but we didn't know exactly from which biological origin it was. And many bivalves can produce this type of nacre, and I show some of them here. Then we had some little clues. Some of these Doppelknöpfe, they have a very tiny layer of prismatic crystals, but not always. It's very tiny. But these prismatic crystals, you see them here. If you make FTIR, infrared spectroscopy on these tiny crystals, you see that they are made of aragonite. So they are not calcite, they are aragonite. And also, we did some stable isotope geochemistry of delta O18 and delta C13, and it gave some negative results, so delta in a negative zone, which suggests a freshwater or estuarine origin. Okay, but then what we did was to do some microextractions of proteins from these buttons. Of course, we didn't sacrifice too many buttons, just few, but enough to have good sampling. And what we found different proteins. So these are really tiny micro extractions. And we found some proteins like HIC74, HIC52, silk mapping. We don't know the function of these proteins, but we know that these are shell proteins. And all these shell proteins are proteins from freshwater bivalves. Freshwater bivalves. Huh? And it means that the idea that these shells were exported from Mediterranean coast to the north or to the east, this idea does not work. And actually, what was the main point is that for making these Doppelknöpfe buttons, nacre buttons, actually the tribes, the population of that time, they used the local freshwater shell resources. No need of long exchanges. It's just local resources for making these button pearls. No use of marine nacre shell. What is important is that the matter, the nacre itself had no value, but what had a certain value was the way of making all these buttons. Starting from a raw material which had no value, you make something which had a high value because of the work that you put on it. And the button pearls were rare and really required an effort. And what is also important is that this technology sort of spread all across Europe. So the resources were local, but the making of this Doppelknöpfer, this way of making, sort of propagate through Europe, and that's the important message. So it means that in our case, the proteomics on shell material was very effective in bringing a response to an unanswered question. So to summarize, and that would be more or less to summarize about shell protein diagenesis. You have the persistence of some shell proteins even after long heating, like 10 days at 100 degrees. These proteins degrade differentially, so you have different degradation patterns. Diagenesis, which is seen by proteomics, is seen by the disappearance of some proteins, also the decrease of the number of identified peptides for a given protein. So the main message is that now we have all the tools to revisit diagenesis of skeletal matrix protein, especially by using proteomics. And also the idea is also to track the diagenetic behavior of each single protein separately, which is a very nice idea. So you can, for a given protein, you can try to see how it will evolve during diagenesis. So this approach is very well suited for archaeological samples. I don't know for very old samples, million-year samples, maybe it's more tricky, but at least we can start with archaeological sample. And that's more or less my last words. Not completely my last words, but almost. I was asked, before I forgot, I was asked by Stefano, give you, and this is the very painful part of the stuff, to give you a sort of exam. Let's call it exam.
It should be up to you. According to what we have been said.
It should be.
It's up to you how to evaluate.
Well,
in the next week.
Because what I understood from Stefano is that I can give you the sort of exam now, and you will have until May to answer. Is it okay with you? Okay. So because I have prepared.
Question.
Yeah. So these are. So I will. So yeah, I have prepared some stuff. So I explain very briefly. I'm sorry for this, for making the atmosphere becoming cold. But so I briefly explain what it is about. There are three questions. I wait until everybody has a. So don't worry, it's not very. I mean, I'm not a skinner, huh? So it's sort of easy going stuff. Well, there are three questions. Well, one is about, if you look at page 2 and 3, it's a small paper. It's a small paper published in Science. It was an insight, so it's a very, very brief paper. And I just ask you to summarize this paper, meaning something like half a page or at max 1 page. And please, try not to use ChatGPT. I will know it. Okay. So this is the first question. First question is the summary, and then you have two questions Yeah, then you have two questions. One is about differences between, let's say, biologically induced mineralization, biologically controlled, so not something not very, very difficult. And the third question is also very, very easy because this is something we have shown you. Well, I will ask you to answer to each of these questions. Well, question 2 can be made in about 20 lines, question 3 in 15 to 20 lines. So feel free and feel okay with that. I will ask you maybe to send me your response probably by email around May, or do I need to fix a date? Or
May the 9th.
9th of May. Okay, so you know more than me. And you can send me on my address. So that's Frédéric.Marin @ oh, sorry. Yeah, yeah, I should
In
have.
the site of FishMed, your email.
Yeah, but maybe I can.
Better.
Yeah.
The paper is always better.
Yeah, just go.
Sì, sì.
Thank you, Frank, for your participating person in this week. Alcuni di voi mi hanno fatto domande, qualche question internal for the student. Okay. Parlo in italiano, sapete benissimo. Il problema dei crediti. Ok. Bisogna che lo chiariamo, per cui mi hanno chiesto.